I'm Tom Hash, back from Miami for a couple of days, and it's my pleasure this morning to introduce to all of you someone who doesn't need an introduction here at the cherub, Mr. V Rajaram, V for Bengaldas. He's a young man. He was much younger when he joined here as an MSc student a few years ago. Found out this morning he's been with us for 8 years. Stealing his life away from him. Anyway, he started here on his MSc work in 2004. Worked with us from 2004 to 2006 on his MSc. Then from 2006 to 2008, he was a bench technician in the AGL. We use the MSc program here as a way of finding good technicians, and it works. So he will follow at the bench. We exploited him mercilessly, training people how to work at the bench, because he's got really good people skills and he's really good at showing people how to do things and getting them to be able to do it themselves. So from 2008 till now, he's been working on his PhD with us, with the CSIR fellowship at JNTU, and he's going to talk to us this morning about things he's been doing with what at that time was next generation sequencing. Now, say it was third generation. The fourth generation is here, but this was, when he got started, this was frontline stuff, and it still is. So let you tell us what you know, please.
Good morning to all of you. Today I am going to present on my PhD topic, creating EST-based markers to facilitate marker-assisted selection for drought tolerance in pearl millet. So already Dr. Hash has introduced me. My supervisor in the university is Dr. M. Lakshman Narasu, and my supervisor in ICRISAT, Dr. Tom Hash. And later on, when Dr. Tom Hash has moved to Africa, then Dr. Oscar has accepted to be my co-supervisor. Today, just a brief what I'm going to present. I'm going to present about my objectives, introduction, and I have clubbed everything, my materials, methods, results, and discussion together so that it will be very concise. And then summary, conclusion, and a few publications that I have got from my work, and also acknowledgements. So coming to the objectives, my objectives are to create pearl millet ESTs from drought stress tissues of pearl millet mapping population parental lines ICMP 841P3 and 863BP2, and to identify primer sequences for the EST-based SSR markers, and to map these EST-SSRs in pearl millet RL populations, and later on annotate all these ESTs and estimate their map position based on the rice genome sequence. We all know pearl millet is a cross-pollinated crop, and it has a diploid genome of 2n equal to 14, and it is drought tolerant and grown in dryland conditions. It is called as poor man's crop because it usually grows in very drought areas where usually all the poor people are mostly there. And it is a dual purpose crop which is grown for both food and fodder. And it is the sixth most important cereal crop after wheat, rice, maize, sorghum, and barley. Now I told it is an orphan crop. It is orphan crop because it has very limited genomic tools. It has limited number of marker resources like the RFLP markers, AFLP, SSRs, SSCP, SNP markers, and so on. And also is limited number of linkage maps and also QTL maps. Few of them have been published by Alves et al., Key et al., Senthilwell et al., Marek et al., Bentley et al. All these have published reported several markers, and there are hardly about 100 SSR markers, mapped SSR markers for this crop. And also several QTLs have been mapped by Yadav et al., Lee et al., Jones et al., Morgan et al. For the disease resistance, and Napoleon et al. For the store yield and quality traits, like so on. So in spite of all these limited resources, there are some very few successful stories, and one among them is the HSB67 improved. This is HSB67 improved variety, which is released in 2005, which is the first public bred product to be released in India, and it is based on the DNA marker selection, and it is developed by Dr. Hash and team. And the citation is Hash et al. 2006 and Hash and Kerwal 2007. Once this variety has been released in India, it is saving nearly $20 million per annum, and the cost of releasing this variety is less than $20 million. But it is saving nearly $20 million every year for the Indian farmers. So as I told earlier, there is a great need to increase the molecular tools and genomic research for this crop, as it is orphan crop. And for doing this, we have employed next generation sequencing technology. We have used next generation sequencing technology because it is very high throughput and it generates large amount of genomic resources or genomic data in very short amount of time. And when we compare the amount of data generated with the cost, the net cost is very less. So we use the next generation sequencing technology. In 2009, at that time the facilities were the loci or 454 sequencing technology, Illumina, and IBR Solid. These are the few technologies available at that time. And one of the successful stories to say how useful the next generation sequencing technology is, recently the PGNP genome sequence was cracked by Dr. Rajavarshini and team, and it has been published in the Nature Biotechnology. So this shows that in very short span of time we can do large amount of sequencing and with very less cost. So we have employed next generation sequencing for generating the data, and the genomic resource data for Millet. So coming to the materials, methods, results and discussion, wherein I have clubbed everything. The objective one. First objective is to create pearl millet ESTs from the drought stressed plants of pearl millet, mammal population parental lines, ICMB 841P3 and 863BP2. The ICMB 841P3, this is sensitive to terminal drought stress, and this is resistant to terminal drought stress. So our objective was to generate ESTs from these tissues because they will help in identifying genes related to drought tolerance. So what we have done was that first we have imposed the drought stress. How we have done was that using the standard drought protocol, which was developed by our physiology group, Dr. Vincent group. So what the stress, we have selected all the two genotypes. This is the ICMB 841P3 in the picture that we are seeing, and the 863BP2. So what we have done was that at the boot leaf stage, we have when 10% of the plants have come to boot leaf stage, we have enclosed the whole pots with the plastic bags and just left an opening at the base of this stem for adding water. And daily the transpiration losses were calculated. And for the stress treated plants, we just added reduced amounts of the whatever the transpiration loss was there. And for the control plants, we just maintained the plants at field capacity. So like that, daily the reduced amount of water was added to stress plants, and there was a slow onset of the stress, like there was slow imposing of the stress on these plants. And we continued this stress treatment till the plants have reached 20% of the normalized transpiration ratio. So once, so as the stress was going on, so we had to do the RNA sampling at 4 different stages. So, and we plan to collect tissues from the leaf and root of the respective genotypes. And we collected tissues at 4 days after initiation of treatment, at 70% normalized transpiration ratio, at 40%, and then at 20%. We have collected we have sampled all the tissues, and then we extracted the total RNA from each of the sample using hot acid phenol methods, Schmidt et al. And after extracting the RNA, we pulled all these 4 samples together, like for this, all these 4 over here and all these 4. And finally, we got 4 samples of total RNA. And then what we have done was that we have synthesized cDNA using the super smart cDNA synthesis kit manufactured by the Clone Tech. And finally, we have generated 4 cDNA samples. Once the cDNA was ready, we have sent this to John Craig Venter Institute, USA, for doing the sequencing using next generation sequencing technology, which is the FLXR 454 sequencing. So all these 4 samples we have sent to them. So what they do for this 454 sequencing is that first they do the fragmentation of the DNA, and they transfer these fragments onto DNA capture beads. And once the fragments are transferred to DNA capture beads, then they do the emulsion PCR followed by clonal amplification, and then next these all the clonally amplified DNA bits are transferred onto the picotiter plate. Once the picotiter plates are ready, then they go for the sequencing using the FLX of 454 sequencing. So since there was constraint of money for our project, they just plan to do half plate of each picotiter plate for sequencing, like half plate for here and half plate here and half plate here. So generally this 454 sequencing with the facilities available as of 2009, you can generate around 400,000 ESTs. Since we had done only half plate, we have got close to 200,000 ESTs for each sample. And once we have got the raw ESTs, what we have done was that we have cleaned all these ESTs of the ribosomal RNA sequences and the poor quality sequences, vector and raptor sequences. And the cleaning was done using a program called as seqclean. Once this cleaning was done, we have got cleaned ESTs of which are about 58K, around 139K, 71K and 127K. There was significant reduction in the number of ESTs for the leaf tissues than the root tissues. This is mainly because since we have imposed drought stress, there was more more drought stress. The leaves were at the drying condition, and there were, I think that was the reason why we got more ribosomal RNA for the leaf than the root tissues. And so we have got lost significant amount of the sequences. And since we removed all the short rates also, the average base pair length also increased a little bit to 240 base pairs. Now once we got all the cleaned ESTs, then what we have done was that at JCI we have assembled using the Planta pipeline, which basically uses a TGCAL, which is nothing, which is also based on CAP3 assembly program. So once this assembly was done using the Planta pipeline, we finally got contigs of 34K and singletons of around 78K, and these together are around 112K, which are called as tentative unique sequences or unigenes. So once the assembly was completed, then we had to develop the markers. So next objective was identify primer sequences for the candidate EST-based markers, EST-based SR markers. So how we develop the markers? So now we had got all the assembly and we have got the all assembly data. So what we have done was that we have used a tool called as MESA microsatellite, which searches for the class 1, which searches for the SRs. We have given parameters for the class 1 SRs, like the repeat length has to be more than 20 base pairs or equal to 20 base pairs, and we identified all the class 1 SRs. Once we identified the class 1 SRs, we used a program called as primer tree program and to design all these primers. And we there all the parameters are standard, but we limited the product size to 150 to 400 base pairs because we wanted to do the analysis on the ABS sequencer, and the limit for it is 500 base pairs. And all the newly designed markers were called as IPES markers because with the abbreviated because of ICRISAT palmate EST stress. Where ICRISAT palmate EST stress is what the expansion stands for. And XIPS refers to the marker locus. So just to give a summary, out of the total 2S 1,12,864 unigenes, we have found around 502 class 1 SRs. Later on we have removed all the redundant SRs as well as some redundant SRs, and some SRs could not be designed because they are SRs towards the end of the sequences. So finally we could design 236 SRs after doing several kinds of analysis. So now apart from these SRs, we also identified around 5,800 SNPs in 2,146 contigs for these two, between these two genotypes. These are also the potential EST-based SNP markers, so for the future work, we have shortlisted nearly around 553 SNPs for future work on the for future assay on the Bead Express or Casper assay. And the basic criteria that we have selected was that to select, we have removed all the spurious or false SNPs based on several criteria, and later on we selected just at one SNP has to be there per contig so that we don't want to have redundancy, and we have selected a maximum of 1 SNP per 0.5 MB interval of sorghum genome, so that we'll since these serials are very closely related, the uniform distribution on sorghum genome will also mean that we are going to have some uniform distribution on pearl millet genome. So that was the criteria, and we have taken the minimum read depth as 4 with a minimum of 2 per each genotype for selecting these 5 SNPs, but this is just for the future sake. And once we have developed the EST-SSR markers, we want to map these polymorphic EST-based SR markers in the pearl millet areal populations. So we are having 4 polymerate RL populations, so we want to map on all these things. The first population, that is ICMB 814P3 cross 863BP2, which is having 107 F7 RLs, and it is one of the major process that we are using in ICRISAT, and it is segregating for several traits, especially the terminal drought tolerance, salinity tolerance, downy mildew resistance, disease resistance, grain yield and grain quality, store yield and quality traits, plant height and flowering density, plant height and grain density. So, and the next cross that we had used was the H77 cross PRLD 2x89-33 with 149 F7 RLs. It is segregating for the seedling heat stress, terminal drought stress, and grain and store yield component and downy mildew resistance. And the third cross is the 81B cross ICMP 451P8, which is actually the this is the world reference mapping population for pearl millet, and it is having 170 RELs, and it is segregating for the plant height, where D1 is the recessive allele and D2 is the dominant allele, D1 for 81B and the D2 for the ICMP 451P8, and is also segregating for the lang panicle bristles, grain and store yield components, rust and downy mildew resistance, and fertility restoration or sterility maintenance for the A1 and A4 cytoplasmic genetic male sterility systems. The fourth population, PT732P2 and P1449- with 130 individuals, is segregating for the plant height, downy mildew resistance, rust resistance, grain and store yield components, and plant height and flowering time of test crosses. And we have all the 4 classes, and we have developed around 236 EST-SSR markers, and among those 139 have amplified when we checked on the parents, and among those 119 were polymorphic for all the parents of between the parents of these 4 crosses. And apart from these newly developed EST-SSR markers, we have also used the markers available in our lab, or they are the ICMP markers of EST-SSR or ICMP markers, and the PSMP and XCTM series, which are the genomic SRs, and as well as few STS markers. In all, we could find that around 125 were amplifying on these parents, and around 90 were polymorphic for at least for one of the other cross. So once we have identified all the polymorphic markers, so we wanted to do the genotyping on these 4 crosses, and we have used the touchdown PCR protocol, and all the sizing was done on the ABI 3730 Excel sequencer machine. And the polymorphic with clear peaks were selected for the genotyping work, and the genotyping of RL progenies was carried out on the 4 ribs. So once the genotyping work was carried out, we have used G Mendel to do the initial linkage grouping and also the locus ordering. We have taken advantage of the bootstrap option that is available in G Mendel, so that we'll have confidence on the orders that we have done, and we have used 25,000 bootstrap iterations to determine the order of our linkage groups. Once we have determined the linkage groups, we have tried to add the unlinked markers using the mapmaker try and build commands. Once we have added all the markers, then we use the record software to do the final order at lot 3 and Halden mapping function. We have used record software mainly because it orders loci very nicely when they are very tightly linked. And even when you use record software, there was no significant change in the order of the loci, but the map distance was a little bit decreased compared to the map maker. So finally, once the linkage maps were constructed, we have visualized all these things using the map chart. So now just I am not showing the maps of all these 4 things, just I am giving a brief summary of all the linkage maps for the 4 crosses. So we have got for 7 linkage groups, they are nicely grouped for the crosses. First for the recombinant inbred population A or ICMB 814P3 cross 863P2, and also we got 7 linkage groups for the second cross. But the linkage group 6 was divided into 3 subgroups for the cross 3rd and also the 4th cross of the RIP-D. And it is we could say that they belong to the linkage groups because of the because of the common markers for the linkage group in this cross, as well as the markers in these groups. And for the RIP-D or the 4th cross, we got one unlinked group which is named as LGA, and it had no common markers with the other crosses, so we could not relate to it as to it belongs to which of these 7 linkage groups. In all, for the first cross we got around 104, we could map 104 markers, and for the second cross we could map 78, and the least was for the 4th cross about 59. And the map length was around 615 centimorgan for the first cross, and the map length was around 690 centimorgan for the second cross. Though the number of markers are more, here the total map length is less, here it is more. This is mainly attributed because of we have added unlinked markers through mapmaker for this linkage for this cross. So just to give a summary of all the markers mapped among these 4 RL crosses. For the 4 crosses, you can clearly see that the maximum number of markers are the newly developed. There are more than 50% of the markers are the newly developed XIPS series EST-SSR markers, and the least are the STS markers. And we found that segregation distortion is more. This is a common phenomenon in palm millet because it is highly cross-pollinated crop and doesn't accept self-pollination. So this is a common phenomenon in palm millet. So once the mapping was completed, we tried to develop a comparative map using the map chart, wherein we tried to draw the lines across the common markers for all the 4 maps for all the 4 crosses. This is just a figure showing the LG2 comparative map, wherein which is clearly showing the the locus order is almost the same. Then we have just drew a Venn diagram, 4-way Venn diagram, which is illustrating all the number of shade markers. There are 2 common markers that are common for all the 4 crosses, and there are also these places showing which are markers which are shaded by 2 crosses, and these are unique to each of the cross, like 34 for the rip A, 22 for rip B, 12 for rip C, and 11 for rip D. So once we have completed mapping and the comparative mapping, we wanted to construct a consensus map. So we have used a merge map tool published by V Rajaram 2008, which integrates the component maps to construct a consensus map. How it does is that first it will take into account all the maps it took into account all the maps for the 4 crosses. Then it will generate a directed acyclic graph for each of the map, and these form the input to the merge map. And this merge map, it processes and it gives the consensus DAG as output. This is just small minimized figure which is showing the consensus DAG, wherein all the markers, which each circle shows one marker, and it has generated a consensus DAG. Later on, it is converted into a consensus map or linearized consensus map using a mean distance approximation method that it by mean distance approximation calculation. And this map consensus map was visualized using the map chart only. So these are the consensus map for the 4 crosses. So now details of the consensus map which is generated based on these 4 crosses. So we did not include the 2 markers, the 2 unlinked markers which I had got in the cross D. So among all these linkage groups, linkage group 6 was having maximum number of markers, followed by linkage group 2, and the total number of loci mapped in this consensus map are 174, and among which 100 are this newly developed EST-SSR marker loci, and the total length of this consensus map is 898 centimorgan. So now just to give a brief, the summary of all the mapping that work we have done. So total markers that were mapped across 4 crosses were 171, and we got 176 loci. It is because around 5 markers were mapping to 2 loci, like they map to one linkage group in one cross and they map to another linkage group in another cross. So that is why we are getting 2 loci for 5 of the markers, and the total mapped markers in consensus map are 169, and the total loci are 174. And later on we had even identified the syntonic relationships of these mapped EST-SSR markers, like the ICMP and IPS series, and we had tried to identify the syntonic relationship to rice, foxtail millet, sorghum and maize and brachypodium genomes, and we found that around 87 markers were syntonic, means they had hits on the one or the other genomes, and around in all they are around 89 loci. Now this is the final objective of mine, which I am currently actually I am doing it. That is to annotate these ESTs and estimate their map positions based upon the rice genome sequence. So in all, I have already earlier told you, just to remind you, that we have got around 1,12,864 unigenes, and we had got 2,146 contigs and 5,800 SNPs. So to just start our annotation work, we have started with this SNP containing contigs. So what we have done was that for doing the annotation work, we were using the BLAST2GO software. And this BLAST2GO software, there are 3 steps in this BLAST2GO software, like the BLAST step, which blasts all the sequences onto the to the databases, and it finds the homologous sequences. And next is the mapping step, where it retrieves the gene ontology terms from the databases based on the BLAST definition. And next is the annotation step, where it determines the self, it determines or sells the reliable functions for the Go terms. So now just to give a brief of the BLAST results. So the cutoff was e power minus 10, and total BLAST sequences were 2146, and we got total hits of around 24k. We had got hits for around 1375, and total sequence without BLAST hits are 771. And if you take into account all the top hits for these sequences, we got maximum number of hits for sorghum bicolor, and next is Ziamase, and next is Areza. Actually, at the time when we were doing the database was not updated with the foxtail millet genome, so that's why we got foxtail millet somewhere over Setaria italica. Now the Go terms, from where they are got, all maximum number of the Go terms were identified from the Uniprot knowledge base, and a few from TIR and other from the GR protein. And the annotation, once the reliable functions were identified, we just prepared a pie chart indicating the functions. Total Go annotations, we have got around 6,000 Go annotations for the total contigs of 1224. Remaining were not annotated, or listed one or the other step. In all, we got around 1815 are related to the biological process, around 1921 are related to the cellular component, and 2220 are related to the molecular function. Just to give a summary of this BLAST2GO results, 2146 contacts were sent for BLAST, and we got hits for 1375 and no hits for 771. Among these 1375, 1272 were mapped, and 103 did not have Go mapping. And for the annotations, 48 did not have annotated. 48 were not annotated among these 1272, and 1224 were annotated. This is a summary of the BLAST2GO results for the 2146 SNP containing contigs. Now I earlier mentioned that the synteny work was done, so I am just telling over here. So we tried to identify the synteny for all the mapped EST-SSR markers using the BLAST search of these full-length EST-SSR markers on the genomes of foxtail millet, Pennisetum glaucum, sorghum, Ziamase and brachypodium. And all we had around 116 EST-SSR markers, of which 17 were ICMP markers, and 1 or 2 are the newly developed EST-SSR markers. So this is just a figure for the LG6 showing the synteny relationship. So this is our polymorphic linkage group 6, which is renamed as Pennisetum glaucum 6. This position is inverted with respect to the position in the consensus map. Now if you clearly see, it is closely related to the 5th chromosome of foxtail millet, and also the 3rd chromosome of the sorghum. And you can clearly see that they are highly conserved. Now just to give a summary of all the synteny work, the Pennisetum glaucum, the polymorphic linkage group 1 is closely related to rice 5th chromosome with 5 hits, and also foxtail millet 3rd chromosome with 11 hits. Similarly, it is closely related to the sorghum 9 and maize 10th chromosome and brachypodium 4th chromosome. Similarly, the polymorphic 2nd chromosome is related to the rice 3 and 10, and also the sorghum 9, foxtail millet 9, and the sorghum 1st chromosome, maize 1 and 5, and also brachypodium 3. So like that, I have highlighted each of the polymorphic chromosome is more or less close to at least one or more of the chromosomes of other cereals. So we try to estimate the physical map positions of all the TUS or unigenes on the 5 serial genomes. What we have followed the approach of using the BLAST sheets, wherein we have done the standalone BLAST of all the 1,12,864 genes on the 5 serial genomes, just like we have done for the EST-SSR markers. So what we have done was that these are the results just of the BLAST hits of all the 1,12,000 unigenes on the 5 serial genomes. We got hits for around 83k unigenes, and we did not get any hits for nearly 28k unigenes. Total hits on the brachypodium were around 42k. Maximum number of hits were on the foxtail millet as expected, and 54k on maize, 44 on rice, and sorghum had the next highest number of hits. So showing that this also shows that pearl millet is very closely related to foxtail millet and then to sorghum. So we just drew a Venn diagram showing the number of shared markers across all these 5 genomes, and we can see that around 37k unigenes are common across all the cereals, saying that they are very highly related. And if you see all the almost maximum number of the unigenes are shared across all the 5 cereal genomes, around 22k are exclusively hit on the foxtail millet. So this is all the blast work medicine physical map positions that I have done. So finally, coming to the summary of the work that I have done, what we have done was that first we have introduced the drought stress on the 2 genotypes, ICMB 841P3 and 863 parent, and we isolated the total RNA from the stress tissues, and then later on we synthesized the cDNAs, and later on we did the assembly using the next generation sequencing technology FLXR454, and we got around 1,12,864 unigenes, and from this we developed around 236 EST-SSRs, and 5800 SNPs were identified in the 2146 contigs, and we constructed 4 new linkage maps for the 4 RL crosses and developed a consensus map containing 174 marker loci, which includes around 100 newly developed SR markers. The annotation work of the 2146 contigs is completed, and we try to identify the map positions of these EST-SSRs on the 5 serial genomes. Currently we are carrying out the annotation for the remaining unigenes. Coming to the conclusions, from this work, large amount of transcriptomic data has been generated, and we have generated around 1,12,864 unigenes, and we have developed about 236 EST-SSR markers, of which 139 have amplified, and we have identified around 5800 SNPs. 4 new SR linkage maps have been developed, and this is the second consensus map constructed till now in pearl millet, and this is the consensus map having the maximum number of SR markers reported till date for this crop. And this consensus map can be used as a reference genetic map. This study has increased the mapped SR markers by more than 50%, and fill in the gaps in the earlier pearl millet linkage maps, and this has increased the options for the foreground selections for the marker-straight backcrossing programs in pearl millet. This study will greatly facilitate SR-based QTL mapping and applied marker-straight selection programs for drought stress as well as other targeted traits. The annotation and comparative mapping currently being done would help in identifying candidate genes for the drought tolerance, as well as also help in understanding several evolutionary relationship between the grasses. So from my work, I could get a few of the publications, the first one is the development of a molecular linkage map of pearl millet integrating dart and SR markers, published by Supriya et al., in which they have used the SR mark and integrated the dart markers. The next is the integration of gene-based markers in pearl millet genetic map for identification of candidate genes underlying drought tolerance, QTL, and this work was done by Sehgal et al., wherein they have used some of my marker data. And the next one is water saving traits co-map with the major drought tolerance QTL in pearl millet, where they have used the integrated dart and SR map for identifying the QTLs, published with Pulav et al. Currently, we have written a manuscript construction of consensus linkage map in pearl millet based on 4 RL mopping populations in pearl millet. So this is ready for submission to BMC Genomics. So these are the 4 publications that I have got. I have attended 2 conferences. One is the one in the PAC conference, wherein I presented a poster and submitted an abstract for development of EST resource in pearl millet, and they're using development and mapping of EST-SSRs in 4 RL populations. And the other work was we have presented a poster and submitted an abstract at the National Symposium in Genomics and Crop Improvement at Angro, wherein we published that mapping EST-SSRs developed from ESTs of stressed root and leaf tissues in 4 RL mapping populations. I even attended a training program at Kesargarh CPCRI CPCR Kesargad on Perl scripting for genome analysis, wherein I have learned little bit of Perl scripting. I like to acknowledge my supervisor Dr. Tom Hash for coordinating all this work and supervising me all through, actually from my master's onwards, and Dr. Oscar for accepting me as his accepting to do post supervisorship for me after Dr. Hash has left for Africa. And I am very grateful to Dr. Lakshmi Narsu, who was always helpful to me in the university. And I am grateful to the other scientific staff at ICRISAT, Dr. Rajiv Varshni, for taking care of all the sequencing work and also the assembly and initial analysis work for this work. And also Dr. Vincent for helping me in the stress treatments in the greenhouses, and Dr. Napoleon for helping me in all the mapping, for doing the mapping and consensus mapping work. And Dr. Senthilvel, Rakesh and Trushar, they are involved in different parts of the work. Dr. Trushar has especially helped me doing the consensus map construction. And Dr. Ramo and Santosh for giving me several inputs and advices during my PhD program. I like to thank all my friends, colleagues, staff, technicians, scientific officers, visiting scientists, research associates, and all the people in the applied genomics lab, genotyping services lab, bioinformatics, biometrics, and learning systems unit, crop physiology and breeding unit. I would like to thank the funding resource for this project, the CGS Generation Challenge Program. And finally, I am very grateful to the Council of Scientific and Industrial Research of India for providing me the JRF fellowship for the first years and later on SRF from the third year onwards. So thank you all.